脑人言。
大家上午好，非常高兴这次来参加我们脑人研的这个 talk。今天很高兴邀请到我的好朋友杨婉楠。其实我很少叫她中文名字，一般都叫维尼。维尼她跟温妮她现在是 PhD candidate，依然是 PhD candidate，但已经有了一系列非常重要的发现。那么今天他将跟我们 share 这份工作呢，是他在研究生期间发表在 Science 上的一篇关于记忆的选择性巩固的神经机制。那么我首先介绍一下他的一些个人的情况。皖南是在2014到2019年在英国的爱丁堡大学攻读的 Computational Neuroscience。然后在2017到2018年，在美国的 MIT 的 K-TIE Lab 做一些生物相关的研究。然后之后呢，是2019年，到现在是在美国纽约大学的 Neureport58，就是大家可能一般做就 brain state 的都知道 Neureport58是非常出名的一个神经生物学家，攻读 PhD in computational neuroscience。那他这次的介绍的工作是将生物和 computation 有机的结合起来，发现了这个海马的尖波涟漪（Sharp-wave ripple） 在那个记忆的选择性巩固中的一个贡献。那么除此之外呢，皖南还有一些其他的兴趣点，包括 memory consolidation。Reinforcement learning 和 spatial representation，emotion regulation。除此之外呢，它还有其他的一些scholarship，包括Armagen Scholarship Principals Go Abroad Award。那么在我心目中，皖南是一个非常非常优秀的 PhD candidate，他比我当年强太多了。那么今天呢，非常有幸邀请到他给我们介绍发表在 Science 上的一篇工作。那么时间有限呢，我们把舞台交给皖南。
首先，非常感谢学姐的介绍，I'm very glad to be here. Today I'm going to share my work, study the selective memory consolidation by hippocampal sharp-wave ripples. I guess many of the audience might already know, but for those who are not experts or working in the field of hippocampus, I just want to briefly introduce that in hippocampus, there's this very interesting and perhaps one of the most important finding called place cell. So it was discovered by John O'Keefe, who won the Nobel Prize for this finding. And what he found is there are cells in the hippocampus that are very selectively fired at specific location in the environment. And he named those cells place cells, which led to this hypothesis that hippocampus viewed an internal cognitive map of the external environment. The second very interesting phenomenon in the hippocampus is this events called Sharp-wave ripples. Sharp-wave ripples are essentially population neuronal bursts. They happens in two different states, one in quiet awake and also during sleep. And it's a very conserved phenomenon across different species, and has since its discovery, the sharp-wave ripples have been proposed hypothesis to be linked to memory consolidation. So what is the link between sleep and memory? The hypothesis that sleep serves memory consolidation is conceptually rooted in this so-called two-stage memory formation model. It proposed that memories are initially encoded into a fast-learning store, which is the hippocampus. What makes hippocampus a perfect candidate for this first stage of memory storage is its capacity to form very rapid associations, even in single attempt. So we can perform one-trial learning. But memories at this stage are very unstable and vulnerable to influence by newly encoded information. This is a very phenomenal called catastrophic interference. So one stage is not enough. That's why we need the second stage. The second stage during sleep, essentially, they hypothesize that during sleep, some of the new memories are gradually integrated into the slowly learning long-term store, which is the neocortex. And those that are not consolidated during the stage 2 are potentially being forgotten to free up space for encoding of new information. So the keyword here again is that only some of the memories are stored. So that is the link between sleep and memory consolidation in a very simplified terms. What about the link between hippocampus and ripples? What is the link between memory and ripples? So I want to walk you through what is known so far to give you some important background information to understand our results. So I will summarize what is known about ripples and their roles in memory consolidation for those in the audience that are unfamiliar. As illustrated in this diagram, while rodents navigate in the maze, the memory of this experience is actually encoded through sequential firing of cells in the hippocampus. During sleep, there are events called ripples that take place every now and then. The cool thing is that during some of those ripples, the same sequence that encoded during learning are reactivated again. You can see the same sequence essentially fire again, reactive again in a compressed manner. And when this happens, we call those events replays. So ripples are population events where replays take place. Many of us know that sleep is not only unavoidable, but also important. Among many of the crucial tasks during sleep, there is this one on the agenda, which is to create lasting memories based on what we experience while awake. But what we do not, but we do not remember everything we experience. We forget some of them. Only some of the experience are selectively consolidated during sleep. So the crucial question that remains unanswered is, how does the brain select what to consolidate during sleep? So our insight is that since not all memories during the day are remembered, memories need to be somehow selected during or shortly after learning. This selection mechanism was previously unknown. We hypothesized that awake ripples select the memories, and the sleep ripples consolidate those memories later. This is our study in a nutshell. So how do we study this? Let me start with introduce you to the task. Animals are trained and recorded to perform a memory-based task where the animals alternate left and right from the blue spot here at the beginning, and they make choice at here to go at the left or right, and if they successfully remember what they did in the previously trial and go to the opposite direction, they will get rewarded here. So we record the neural activity of CA1 region of hippocampus while animals run many trials. And here we just plotted animals trial by trial behavior trajectory in one example session. And this trial are color-coded. After learning, we record the hippocampal activity during sleep. Since each trial can be thought of an individual event, so to begin, we can ask the following question. How can we differentiate those different events? So this is the part one of what we're trying to understand, which is, can we really reliably differentiate the individual events? And just to orient you, so the link between this and the fundamental question that we want to answer, which is, how does the brain select what to consolidate during sleep? The link here is that in order to study what is selectively consolidated during sleep, we need to have a reliably handle to first distinguish those different events. And if we can differentiate them, then we can see how they're selectively consolidated during sleep later. So that's our first mission. Can they be reliably differentiated? So if we look at this one individual cell, you can see that this neuron actually have two-place field. You might think, oh, it has two-place field, but if you take a closer look, its firing rate also, color-coded by trial number. You will notice that this cell is doing something very interestingly. So it preferred to fires at the middle of the track during early trials, which is colored in those cold blue colors. And then it actually switched its place field towards the end of the track during late trials, which colored it in red here. So this neuron kind of differentiates different events, early versus late trials. It switched its place field. That's how it differentiates different trials. And this neuron is doing something different here. So it actually only fired during late trials and remained more or less silent during early trials. And this same cell on a different part of the maze behave quite differently. So it fires consistently both during early and late trials, but it has significantly higher firing rate during early trials and lower firing rate during late trials. So we do see those very interesting patterns that suggest that we can actually, as individual cells differentiate its firing rate depending on the events. So in effect,
can
yeah.
a question. So here the position, yes. They're from 0 to 200, right? So I know that
yes.
left is the left arm, right is right. But where
yes.
does the 0 starts?
Yes, sorry. The 0 is, if we go back to a 2D plot, we will see. So here is 0.
So
here is 0. It's colored in blue. So this is where animal is confined between trials, and animals are confined here, because this is a memory task. So we confine the animal there for about 5 to 10 seconds to increase the difficulty. So they have to remember, right, what happens in the last trial, in order to successfully make a decision here. Whether to turn left or right.
Oh, so so the 0
so
means
the turning
bottom.
point is
So
about 100 centimeter. Sorry.
Okay, so 0 is the bottom and 200 is the top.
So 0 is here and 100 is about here. And this is like from 0 to 100 for the left. Sorry, 0 to 200 for the left, and here's 0 to 200 for the right.
Okay, thank you. Thank you for clarification.
Of course. Yeah, by the way, like at any point, if you have any question, feel free to ask. I will try to keep it informal and don't be afraid to interrupt.
So if when you put the animal in the starting point, if the animal turn left and go the counterclockwise,
right.
So
the criteria is it just have to alternate. So it has just go to the opposite arm, that it has to previously visited. And if that's the case, it will get rewarded. But if it fails to do so, if it repeats to the same site, it will not get rewarded.
Okay, got it. Thank you.
You're welcome. So yeah, that was the task. It's a very simple task, and I just talk about that. We have some evidence that this individual cells can differentiate those different events, and in fact, this is not the first evidence. Actually, this kind of thinking is inspired by this early study that I was part of. So the leading author, Chen Sun, and he is both my best friend in science, but also my partner in life, as he's my husband. So he led this study where he designed a task. Every 4 trials, the animals will get rewarded at this box here, and using calcium image, which is a very different technique for comparing to what we use here. And he found that individual hippocampal cells encode event identity. So, for example, this lab1 cells here, they only fire at lab1 but remain silent during lab 2, 3, and 4, even though the animals visit the same spot during the following labs. So he find there are cells, tile this whole space and tile those whole combination of lab 1, 2, 3, 4. Interestingly, those event identity encoding cells are also preserved across days. So lab 1 cells on day 1 are remain to be lab 2 cells on day 2. And this event identity coding is preserved across different environments. So this is very important even if you introduce the animal to a very different environment, when the place code actually remapped, but this kind of event identity code preserved. So back to our story. The picture here is that at first glance, the neural sequences from trial to trial may seem to be very similar, but they are not quite the same. So for example, this kind of example cell, it fires a lot at the beginning trials, but became silent at late trials, decrease its firing rate, not completely silent during late trials. And there's another cell, fires at the middle of the track during early trials, but switch its place field to end of the track during late trials. And this another cell, for example, is very silent at the beginning of the trial, but become very active at the end of the trial. So you get the idea now. Individual neurons show varying firing rate across trials. So the question is, if we have a lot of those neurons, and if we have 4 different combinations of firing patterns, then those unique combinations among large population of cells may be enough to reliably differentiate each individual event. But we know that, distinguish between 4 events is simpler, but, what we want here is to distinguish like 70 events. And we know that signal from individual neuron is fundamentally noisy. This motivates us to harness the power of population analysis. So
another question. So
yes.
If you enlarge, for example, change the size of the maze,
Yes.
would the early neuron always, how to say, fire at the same place, or will fire still at the early phase? So it's time dependent or position dependent.
Yeah, that's a very good question. We didn't do this experiment here. But Chen, in his paper, he did exactly this experiment. He elongated the maze. And what he found is when you elongate the maze, the place field elongated as well. But in terms of, the trial identity, the trial one, like lab 1, in his terms, lab 1 cells remain to be lab 1 after elongation. And the most striking one is another kind of distortion. Like he instead of elongation, he changed the geometry of the maze, right, from square maze to circle maze. So this geometry of the environment totally changed. So in this case, then the spatial code, so the firing field totally remapped, like global remap, like to a different location in this new environment. Yet this trial identity remain the same. So if it's lab 1 in this square maze, it's still lab 1 in the circle maze. So this seems to be a generalizable signal across different environments, unlike the place signal, which undergo remap.
Okay, so I remember if this, for example, the arena is too small, you couldn't reveal the place field of one cell, right? It must be large enough.
Depending on where you record. So if it's dorsal hippocampus, usually the place field can be smaller. But in general, yeah, if you want to have a lot of cells with good place field, you want to have a large enough arena. Yeah.
Thank you. Thank you.
Okay, so let's move on. So yeah, so we want to have a population readout for the trial identity. So what we did is dimensionality reduction. So briefly, we take the neural data while animal running on this we call figure 8 maze, and we can record about up to 500 cells simultaneously while animal performed the task. We bing and smooth the data, and then further did some pre-process to build a spiking matrix like this. Note that since we have 500 neurons, their activity live in a 500 dimensional space, which is a very high dimensional space. So in order to visualize activity in such high dimensional data and find the structure inside, we need to do dimensionality reduction. So here we used UMAP. What does the manifold look like? And this is the structure that after we do dimensionality reduction, at first glance, you can already see that, okay, this manifold seems to preserve some of the structure of the maze. We can even get a closer look at this neural manifold. We correlate features of the manifold with various other variables. For example, we can color the manifolds based on linearized position of the animal. Just to orient you again, this blue is the beginning, where animal confined. At the beginning of each trial, this is where animal turned, and this is when it got back. And you can see that this manifold in the brain literally mimic this geometry of the environment. So this is pretty cool. But this is also expected given that there are place cells in the hippocampus. So you're expected to see, a representation of the space inside the hippocampus, given that there are place cells. So what is even more interesting and a little bit unexpected for me is that when we color the manifold by the trial number, we can see an unexpected pattern emerge. The neural representation not only tracks different locations as animals learn in the maze, it also tracks the progression of the trial events. Now you can see this very structured activity pattern from early to late trials, while where the representation of events that are closer together. So here the color map is in trials, and the cold colors are early trials, and the hot colors are late trials. You can see the trials that are closer in time are also embedded closer together in a neural state space. So that's cool. We basically got a population readout of the trial event identity. this it always strikes me as a very interesting phenomenon. We know that this from Heraclitus, no man ever steps in the same river twice. For it's not the same river, and he's not the same man. And same for the brain. The neuroactivity never remain the same twice. So now we want to move beyond visualization and to actually quantify, right? To quantify this phenomenon. And one way to quantify this is to do decoding. So using semi-supervised dimensionality reduction with UMAP, we get neuroactivity in low-dimensional space. We then train a KNN, which is k-nearest neighbor decoder, and further validate the coding result with tenfold cross-validation, which allows us to visualize the cross-validation result in a confusion matrix. Just to orient you here, you can see that the probability concentration really concentrated on this diagonal, which means the predicted trial, it matches the actual trial, and which means that we can reliably decode or predict just from the neural data which trial is this, with very, very high precision. I also want to emphasize that, UMAP is a very great visualization tool that gave us a lot of intuition to think about high-dimensional data, but it carries with its own limitations. Most notably, it can introduce some distortions. During this dimensionality reduction process, it tries as hard as it can to preserve the structure in original dimensional space, but inevitably there might be some distortions. So we were very careful. We don't want to just rely on UMAP. So we further confirmed our result with 4 different results, including PCA, principal component analysis, and Bayesian decoding, which is very common in standard decoding tool in hippocampal physiology. We also used decoding from the original high-dimensional space, both using Euclidean distance as the distance metric, also use cosine similarity as distance metric. Those are technical details that it's probably not that important, but the crucial message here is that we confirmed with different method and we got the same conclusion that trials identity can be very reliably decoded with high accuracy from the population activity. So here I just wanted to digress a little bit and kind of shout out to this little website that I made. And if anyone in the audience that is interested in applying manifold analysis to your own data or doing decoding, and if you want to get started on this, you can check out the demo code and tutorials, which is available on our website that I built. I don't know if you can see. Do you see the slides or the website?
It's a website.
Okay, yeah, cool. So yeah, this is the website. And if you click on this results and demo session, and if you click this first part, and this is where I just intuitively introduce the result again, like much informal comparing to the paper. But also here you have visualizations that are interactive, so you can actually drag and rotate this manifold. Kind of, I think, very good for you to gain intuition about the manifold and about the data. And most importantly, you can click here where it will lead you to the code that you can basically replicate what we have in the paper. You don't have to know anything about Python or Python environment, so you just click through all those cells. You can follow everything in this step-by-step tutorial. and you load the data. Basically, what you can is you replicate the process of how we deal with the behavioral data, the electrophysiology data, and in the end, if you scroll down, you will be able to reproduce the result that you saw in the paper. So if you want to get to know about the method a bit more to get some hands-on experience, I recommend check out this tutorial, or tell your friends who might be interested in this. I spent some time to work on it. But back to the presentation. Okay. So that's kind of the part one. We answered this question that yes, we can reliably differentiate different events. Hippocampal population dynamics can differentiate different trial events, and we confirmed we can reliably do so with 5 different methods. And just to remind you again, the reason why we did this exercise is that we want to answer this fundamental question of how brain select what events to consolidate. So we have to first differentiate those events, and that's the part one. Now move on to the second part, which is regarding kind of the robustness and the universality of the phenomenon that I just introduced. This part will be a little bit technical. For the interest of time, I don't want to go into too much detail, but I think it's important to at least show you the gist of it, because this is the part that we did for control, thinking about all of the alternative things that could happen, just to make sure that we have a solid ground before we move forward to look at sleep memory consolidation. But I will try to be as brief as possible as well, just to communicate the essence. So first of all, we just wanted to make sure that the phenomenon that we recovered is a structured, this variation between trials is a structured change rather than a random fluctuation in the data. You can have two scenarios. One is you have very structured variation between trials that allow you to differentiate between different events. The other hypothesis, you just have some random noise in your data, so that this random noise is enough to drive the difference between trials, and that's why you can decode different trials. So those are two valid hypotheses, and we wanted to make sure that our data, it's a structured change rather than random fluctuation. And to do so, we did a simulation with Poisson process. So we simulate individual cells based on the property of the real cells, and then we pass on those simulated population of cells and build a manifold based on that. And we confirm that the simulate data, which is in the bottom here, it can recapitulate this position-driven manifold structure just as the real data. But the simulating data, which based on random fluctuation, could not recapitulate this structural change across trials. So this gave us confidence, this is decoding result. This gave us confidence that our phenomenon cannot be explained away by random fluctuation. That's the key message. And the second thing, which this is a question that I got asked a lot when I present, the reviewer also asked, so how can we make sure that the representation drift that we saw it is not actually due to a much trivial explanation, which is a probe drift. So essentially, you didn't see real change that happens in this data, but rather you didn't do experiment well. Your recording is not stable, and there's electrode drift So that's why you see variation across trials. So that's a very, very important kind of scenario that we wanted to exclude in order to build on a solid foundation. So we did some very careful analysis to make sure that there are minimal probe drift as measured by centroid displacement. And we also did some other control experiment to show that this variation or that could potentially introduced by probe drift cannot explain away our funding. And next thing is, could be interesting for the audience. So basically, we confirmed that this event identity encoding arises from a distributed population code, which means that, it's not like individual cells is enough to give you decodability for the event identity. So basically what I did is I subsample. So I take the data, which, we record 500 cells simultaneously, but then I subsampled, I take away some of the cells, and I did subsamples of different sizes from, I only take 50 cells from all the cells we recorded, or all the way to 450 cells. And this exercise, you just want to see, if you have less cells, then you wouldn't be able to see this structure that we see. This very structured change across trials only emerge if you have the power to record from large population of cells.
Maybe I missed. So for the 70 trials, it's within one day or it's across several days?
So this is one example session. There's variations between days just because sometimes animals are not as motivated. So this is just one example session.
So I know, as you mentioned before, you give like water reward, right? To train the animal.
Right.
But I think after hundreds of trials, after several trials, animal may be adapted to the trail. I don't know how to adapt it to the environment.
Yeah.
The memory disappeared, something like that.
Memory disappeared. So,
like, you don't need to remember the environment purposefully.
You don't have to remember the environment. You only need to remember what you did in the past trial, right? You always have to remember what you did in the last trial. Even if it's the 70th trial, you still need to remember, where did I turn during trial 69, right? So it's because it's the memory part not coming from familiarity with the environment, but with its own previous action. Yeah. Okay. So it's not depending on.
Is there a criteria that the last trial must be turned left and next be right? Is there some criteria? Is there some criteria for the turning directions for different trials? So why does animal need to remember his behavior during the last trial?
Oh, if it doesn't go to the opposite side, it will not get rewarded.
Okay. Maybe I missed that part. Okay. Thank you.
Yes. Yeah, and the next is that we checked if this phenomenon is observed in different tasks and rodent species. This is kind of answering the universality question. So we don't want it to show a phenomenon that's only happens in one specific task. So we checked the future animal to run on a linear maze. We still see the same phenomenon. We see the trial by trial variation very reliably encoded in the population data. And in a very different maze, in this radio arm maze, we also see this trial by trial variations very reliably encoded. So we conclude that this phenomenon is universal. across different species from mouse to rat and across different tasks. Lastly, I just wanted to say that, beyond those tasks, there's a very, very different task now that's like an auditory guided task where, I don't have time to go into the details, but a very, very different task. We use the similar method, dimensionality reduction, to kind of uncover the representation there. For the interest of time, again, I don't have time to tell you about this amazing study, but it's really has some very ingenious experimental design and asked a very fundamental question in hippocampal representation. It's available on archives, so if you're interested, you can go and check. And the good news is it has been just accepted in Nature. So you will be able to see this paper hopefully soon. So that's part 2. Let's just summarize what I have shown you so far. Event-specific population representation is structured, and it cannot be explained by probe drift. This different event coding is preserved across diverse ranges of tasks and rodent species. And going back to the fundamental question we want to answer at the beginning of the presentation, how does the brain select what to consolidate during sleep? Now, finally, after this, we can move on to this most important question, which is selection and consolidation of events by ripples. But before I tell you about how we did it, I just want to tell you how in the past people have done this and why this failed, why this old paradigm failed. So in the past, when people wanted to study how memories are replayed during sleep, it's a very hard question. During sleep, there is no behavior readout. So how do you know what is encoded in the neural activity? So in the past, people typically use Bayesian decoding. The intuition is that you build a template when animal is awake. You sort the cells by the spatial location, and then during sleep, you just match your activity recorded during sleep to that template, and depending on where it matched, you can decode where it's being replayed. But the problem in the old paradigm is that you build this template, you need to have the average firing rate across trials. It's very data hungry. You need to average across trials. But that is not working for us because we want to study the replay of individual events, not the average across all the events. So that's why we need a totally new, different method. So first of all, we need new technology that allow us to record from a lot of neurons simultaneously, which will give us the power to build the manifolds that differentiate with very high granularity between different events. Secondly, we need this new kind of analysis method to allow us to study this question. So move on to the result. So I mentioned earlier that ripples happens both during awake and during sleep. So first we can look at awake ripples. So this is one example awake replay event. So we can see that, for example, in this event, animal is here, which is where it get rewarded. And while it is here, the replays in the hippocampus actually replay this part of the maze. So this is kind of the trajectory where it will go later. And this is decoding from awake replay. And this has been done in the past, even though using different method. But what is really cool with our new method is that we can not only decode where is replayed, we can also decode which specific event is being replayed. So in this example event, the animal is here and it's replaying this like trial 1, 2, 3, 4, 5, 6, 7 trial block number 7, for example. So this is just an example event to give you some intuition. So we ask this question: at a selected trial, does the awake ripples tend to represent or replay the past, future, or current trial? So our analysis suggests that most of the time, awake ripples replay the current trial instead of past or future trials, even though there are some exceptions, but most of the time it is consolidating the past trial. And the next question we want to ask really is linking between the awake ripples to sleep ripples, right? We saw the awake ripples that mostly replay the current trial. How about sleep ripples? So this is an example awake ripple, and on panel D and E is an example sleep ripple. You can see from the raster, like it's this yellow population of cells firing is very similar firing sequence. It's decoded to be replayed not only in a similar spatial location, but similar trials. So this is just one example to show you the similarity of some of the sleep replay events with the awake replay events. But we wanted to do some summarize analysis across all the events, and here basically is the most interesting thing that we find. If we plot the distribution of all the replay events during sleep, and if we look at like which trials are being replayed during sleep, we can get a distribution like this. Basically, in this is one example session. In this particular session, so those late trials are replayed a lot during sleep, and these early trials are like not replayed at all in this particular session. And if we superimpose the distribution of maze replays, we can see this amazing pattern. That's really kind of the most important result in this paper. We see this very striking mirroring correlation between what is replayed during awake replay and what is further replayed during sleep much later. To do some control, because there's also pre-play phenomenon, which is replayed prior to the experience. So we did some careful control, make sure that this does not occur for pre-play. So this phenomenon only occurs, this correlation only exists between post-sleep replay and the maze, suggests that this is a learning induced change, not a priorly existing phenomenon. These are just more example sessions, and we also did a summary across all the sessions and all the animals. We see this very, very high correlation between awake's replay distribution pattern and the post-sleep distribution pattern. Furthermore, we use a mixed effect linear regression analysis. And this analysis is essentially just tell you, among all the factors that could possibly predict what is replayed during post-sleep, which variable is the most reliable variable. across different methods, again, we use different methods to test our hypothesis, and across all of them, maze replay pattern best predict what is happening during post-sleep. This is perhaps not important. Some details we confirmed the consistency between different trials and consistency between different decoding methods. You know, if different methods decode the same event as the same trial. And we confirm it is indeed the case if you have a lot of cells, but if you have less cells, you wouldn't get consistent results between methods. So just to summarize, we isolate a possible neuromechanism for how memory selection works between awake and sleep. So our hypothesis was that awake ripples selectively replace some of the events, and those events that are selected by awake ripples that are, has the chance to be consolidated during awake are then prioritized to be replayed much more often during sleep and get potentially consolidated. So the take-home message is that awake ripples, we propose that awake ripples act as a memory tag, such that those memories that are tagged by awake replays during or shortly after learning are selected. They're special. So then later during sleep, those selected memories are consolidated many times through replays during sleep. And potentially those events that are selected and consolidated much more are incorporated into the existing knowledge structure, and then the non-selected memories are forgotten. So this is our working hypothesis supported by the data. Okay, I hope there are lots of ripples take place inside the brains of the audience now, so that you can remember what I tell you today. But that's all. Thank you very much.